So when we talk about unculturable bacteria, BBNC was one of the terms, right? We were talking about the possibilities why some bacteria, even if they are present, they are alive, they are active, are not seen in the laboratory cultures. They cannot be isolated in the laboratory conditions. That is what we were discussing, right? So in that context, we have talked about various possibilities, and one of the thing was like maybe the organisms are dormant, but what is that dormancy does mean? Is it just formation of spore? It is one, spore formation is one of the basic mechanisms which is shown or depicted by some of the bacteria. All the bacteria, they do not show sporulation, right? So whenever we are talking about the bacterial mechanisms to cope up with the surrounding and the changing environmental conditions which create stress, right? Bacteria under such stress conditions are not able to multiply, not able to grow, not able to form the colonies, but they are surviving, right? So when does really this kind of situation would happen? So a schematic representation I'm sharing here that shows the difference between various you can say the uh, phases that cell would go through if it is challenged with various stress conditions. So actively growing cell, if it comes across certain physical stress conditions or unfavorable conditions, then it may go into some dormant phase, and in that dormant phase it will try to run away from the stress, but at the same time it is alive. It is alive, and as the favorable conditions resume, it will resume the growth, right? So in this diagram, if you are saying this is a schematic diagram that shows the viable cell, so it is actively growing. When the growing cell we talk about, we know what are the growth phases: lag phase, then exponential growth phase, then stationary phase, and then the decline, right? Meanwhile, during exponential growth phase, when we talk about, we get duplication of cells. So regrowth is one of the important um, facets when we talk about common growth characteristics of a bacterial cell. So that phase is lag, exponential, and stationary phases. They depict the culturable phases, right? In even if the cell is in the stationary growth phase, when you are putting it onto the media, it will show you colony formation. It will grow on the media, provided the media conditions are appropriate. Now, when it is exposed to stress conditions, it shifts from culturable to unculturable. So it is not able to grow under such conditions, but it is going into some dormant phase. It is unable to divide. So VBNCs is one of such kinds. Viable but not a culturable bacteria. They are not going to grow. So it is a kind of adaptive strategy shown by bacteria to survive in extreme environmental conditions. So always not sporulating. I mean, sporulation is a kind of a blessing that some of the bacteria they have and others they do not have the genes required for that. So do not they get a chance to run away from extreme conditions and experience the dormancy? Yes, they do have, and that is what is the phase which is referred to as VBNC, that is viable but not culturable. So the stress extreme conditions may be like nutrient starvation, increased salinity, pH changes, osmotic stress, oxidizing active oxygen species, or maybe UV rays or even white light in certain cases, or application of certain uh, anti antibacterial agents, maybe chemicals or physical ones, right? But are these VBNCs are not active, right? They are inactive, but are they alive? How we get to know that they are alive? They are alive because their respiratory activities and their metabolic activities are happening, but at a very, very, very slow rate, right? Why this happens like that? Because there are changes happening in the cell membrane, peptidoglycan layers, then lot of ATP levels are there, increased ATP levels are present in such kind of cells, and the membrane is intact. It is not injured, it is not broken, but it is altered in a way that membrane activities are compromised. So, some kind of molecules which are deposited will make it uh, comparatively non functional to the rate otherwise the cells do their activities, metabolic processes. Of course, a good metabolic activity is a sign of very flexible and selectively permeable cell membrane that we all know. So there are changes in the cell membrane. Does it have complete and non-destroyed membrane? Is it um, uh, like injured? No, not at all. It is safe. It is integrate membrane, but it is having compromised functions. Secondly, what about RNA and DNA? Has it lost its any of the RNAs or DNA as it happens when the cells are challenged with UV, etc.? No, it is not like that. Or when the cell goes in the dead phase, generally membranes become leaky. Some of the DNA is released outside, and step by step, all the functions come to an end. So no, VBNCs, they do not show any of these characteristics. They have their RNA, they have their DNA, they have very high amount of ATP levels, they have respiratory and metabolic activities, but at a compromised rate. So, provided an opportunity is given, they can resume their growth cycle again. Alright. So, such VBNCs, if they are present in a given environmental sample, they will not be easily growing in the media unless the media conditions uh, are conducive for their growth. Right. So, this is one of the things that needs to be taken care of in addition to various things that we were talking. How are we saying there are some certain bacteria are there but they are unculturable? What makes them unculturable? And can they be brought into culture? What efforts are to be taken, so on and so forth. So certain points we had discussed yesterday, today we are continuing with that. So when we talk about various cultivation approaches, so this is what we had ended our lecture yesterday with. So you're going with mimicking the natural environment. Diffusion chamber is one of such design that is built up to promote growth of unculturable bacteria, and n number of experiments have been done with these diffusion chambers. Then soil substrate membrane systems and eye chip and magnetic nanoparticles mediated isolation, etc. So these we have to learn one by one today. So when we are talking about soil sample, for example, a terrestrial subsurface, if you go for sampling, you will find 2500 into 10 to 26 microbial cells if it is active environment in the soil or a rich and healthy soil you are talking about. More than 70% of these are uncultured. So, physiological and ecological impacts remain largely mysterious. We are not able to talk about who is doing, how these processes are happening, which bacteria are responsible for that. Can we use them as inoculants for soil health, or bioremediation of certain xenobiotic compounds which are added during man activities, and so on and so forth. All this remains unanswered because we are not able to see them, we are not able to grow them, we are not able to study their properties, etc. So yesterday we were talking, yes, we can identify them because molecular tools are available, and metagenomic studies and molecular data can help us to identify the microbial taxa of bacteria which are not easily cultured. That is what we had ended the talk at yesterday. But yes, instead of having molecular data, instead of having omics approaches, yet we do not really land up with the answer that yes, we can culture, or we can identify, or we can know about the entire flora or microbial flora I'm talking about of a given environment. So for identification of key microbial taxa and critical metabolic processes in a given environment, a complete interpretation of omics based data is still constrained. What is the constraint? Why it is not possible? The very basic thing is we don't know who are they. Otherwise, what we do, we go for culture, purification, isolated pure colony, DNA sequencing, right? 16S rRNA sequencing, blast, and then we get to know what is the percent similarity, and this is this particular taxa, and it is the species of it, and it is the variant of it, and so on and so forth, or it doesn't match with anything else. But if all the things are of this kind, it does not have anything to match with. So you don't have the basic library um, as rich as to compare these isolates for. Which genera shall I place it? Which species shall I call it? Is a later part of the story. But which group I should place it in? Right? You have diverse groups of bacteria also, right? Depending upon their metabolic processes, the environments they come from, and so on and so forth. But if I don't have anything, any record available to it, what I am going to compare the data I obtain? I am having the sequence, but with which sequence, which type of organisms, which source shall I compare? Because all these things are empty. I don't have any data yet deposited because they were not isolated, they were not cultured, they were not purified, and lack of information is one of the concerns. So, genomics and all other omic methodologies are there, but again, they are not foolproof because libraries are not adequate even today. And again, the point comes back, unless they are cultured, unless they are purified, how are you going to go ahead with it? So, people are approaching various cultivation methods and trying to improvise those. For which yesterday we had said ki use of carbon source, or maybe mimicking the environment, natural environment. So, how it is really done? Right. So successful recovery and cultivation of environmental microbes in the laboratory is going to be very big challenge when it comes to unculturables, and it critically depends on appropriate growth media, incubation conditions, ecological habitat parameters, then enrichment culturing. That could be one of the common initial steps. So to selectively grow one type of bacteria which you are interested in, that will outgrow, and you will be able to find it out on solid media and so on. The choice of organic substrate is very much important when you are talking about enrichment medias. All right. So organic substrate, generally what we provide is yeast extract, glucose, acetate, or maybe certain times pyruvic acid or lactic acid, they are added. All right. We think that simple the organic carbon source, better it would be to grow the organisms which would prefer to utilize that carbon source. So glucose is considered as the simplest form that is assimilable by most of the bacteria, and that's why generally the enrichment media they do have glucose. However, when we talk about this routine method of using defined and culture medias having glucose like or acetate like carbon source, uh, hardly 2% of microbes on the earth we are able to identify and use them, cultivate them, and so on. Right? So, what we need to do is to select conditions that will allow. The conditions mean there are only two things: the media and the abiotic factors, and third is the biotic factors. Right? So, is it that instead of using glucose or carbon source as pure as glucose, what if I use a complex carbon source? Is it possible? Yeah, for research purpose, if you want to really quantify how much of particular molecular molecule to what I mean, sorry, what is the quantity of a particular molecule in a given media, you will have to take that complex material which you are using as carbon source and go for its molecular studies with respect to chemical characterization. So maybe NMR, IR, and all other analytical tools you will have to implement, use, and know the chemical structure of composition of that material. So complex media, when I say yes, yeast extract can be said as a complex example of complex media, but yet it is yeast only. It is not as complex as what is there in the soil when a plant is undergoing degradation, or animal is killed and is buried and it is undergoing degradation and is acted upon by n number of bacteria in the soil, its own flora and flora of different insects, and so many metabolites are produced, and then it will be a complex media, right? Or decomposition of plant material similarly. So these are going to be truly complex nutrients. What if we add them to the media? Is it acceptable as microbiologist? Surely not. But these are the things which will take your artificial culture media possibly closer to the natural conditions, and to certain extent you will be able to mimic the true natural conditions. So people have done various efforts in these lines, on these lines. And what are the outcomes? Let's see. So when you are talking about microorganisms to be grown in the laboratory, can we use complex natural uh, substrates, right? For example, DOM is the term used here, which stands for dissolved organic matter. So any water body, may it be well or river, you see there is a clean water surface layer, then clean water layer, then sediment layer, which is generally a very smooth and very fine soil and clay is there, and it is rich in whatever all has got dumped into that water body, ultimately goes and settles at the bottom and degrades under there, under those conditions, right? So that clay which is generated is referred to as dissolved organic matter, right? So a source of dissolved organic matter. So can we use that as the complex nutrient media? So such experiments were done by different researchers to explore the subsurface flora of soil, or maybe water body, or marine systems, etc. Right? So what are the results? Let's see. I'm going to just discuss one experiment what they had done. So what was the idea? Was to find the flora of groundwater sample. So they had collected it from a well, and sample was shipped immediately to the lab after collection. And of course, while transport, you have to see to it that no bacteria is going to get killed or is going to get overgrown. So the temperature should be kept in such a way that all the microbial activity is ceased temporarily. So in the parcel was packed along with the ice packs, stored at 4 degrees Celsius up to 1 week, and then you can ship it, right? You can transport it to the laboratory. So when you are going to sample the particular water body, or you are picking the sample from soil, that time you are supposed to take the basic note of characteristics of that water body or soil sample site, right? So for example, in this case they had recorded the temperature of the water in the well, and it was around 15.4 degree, pH was 6.37. The dissolved oxygen that was reported as 1.39 milligram per liter, which can be highest as much as 8 milligram per liter when water is really really clean, clear and pure, right? TOC was noted, and all these parameters like phosphates kitna hai, and as many parameters they could note, they had noted it down, redox potential, etc., etc. Now with this basic information and sample collected, what is happening at the laboratory scenario? So the water was taken, it was filtered through 0.22 micron pore size filter, and it was put into the culture medium, right? Now what different types of carbon sources they had used? Like they had used oleic acid and cellulose as one of the mm, uh, complex um, carbon source. Another carbon source was DOM, that is dissolved organic matter. That material they had taken. In one another case they had taken uh, I mean, the thing, can you imagine, they had allowed certain bacteria to grow on the plate which were fast growing. The R strategist, they bloomed as they got the media, and they had scrapped that growth, and then they had killed those bacteria and put in a media that was autoclaved, and the organic matter generated from that bacterial colonies was put into the media for the growth of uncultured bacteria, right? Like that also could be source of complex organic content. So the organic content was changing, the sources were different, and they tried to see which of these works well to to isolate the truly new bacteria, or those ones uncultured, right, or unculturable. Now what they had observed was the the photograph shows you the schematic representation for what different types of results they had obtained. So it shows you NMDS. What does NMD stands for? It is non-metric multidimensional scaling. That means a plotter graph is prepared by comparing two um, samples with respect to different variables. For example, two bacterial colonies, if you are taking and you are taking n number of parameters and comparing them, they can be put onto the entire data for all the characteristics you have studied can be put into this particular format. So non-metric multidimensional scaling plot is there, and Bray-Curtis dissimilarities of bacterial community. It is one of the statistical tool which is used when you are talking about ecology and finding out bacterial indices, etc. So that is used here. It is just to introduce to you these two concepts. No one is going to ask you about this in the examination. Okay, write about it, but you need to know okay, what are the different tools, methods are used when you are studying uh, uh, s- cultivation of bacteria, specifically the unculturable ones, or when you are approaching for the new bacteria, how you are going to compare and obtain the data. So in this case, as you are seeing, both C type and length of incubation, that are the two things. Now C type is the one where nutrient complex was obtained by killing other bacteria and putting the extract into the medium, right? That gave the best possible result, and they had incubated the plates for over period of time, say after 48 hours, 72 hours, like that, 1 week, 2 week, 3 weeks, like that. And what they observed is key incubation time has effect, significant effect, right, on uh, the isolation, results of the isolation. So bacterial community in enrichments amended with glucose, acetate, benzoate, etc., they had given a very good bloom in the initial days of incubation, right? And slowly, slowly, that bloom remained, but those new colonies, a new isolate appearing, was very less or scarcely available. But when the media was composed of DOM, right? Dissolved organic matter, or some diverse sources of complex organic material, that time the probability of getting more number of new bacteria, different types of bacteria, was better. So initially the growth was less, but with longer incubation, variety of different types of bacterial isolates they could get. If you put in the media glucose or acetate, like simple nutritional components, then whatever the R type bacteria are strategists, they grow very fast and they overgrow and they crowd the plate, right? But if the organic carbon source is complex, then slowly you will find that the number of bacteria in which are appearing will increase slowly, but over the period of time you will have a good diversity of isolates. That is what is their observation is. So this is one of the strategies, that is you use complex organic matter, and that is obtained from the same source from where you want to isolate the bacteria, right? It can be a marine sediment, it can be for example mangroves, I mean bacteria from mangrove region you want to obtain. So soil which is in the vicinity of mangrove islands. From there you have to have the organic matter debris. Of course that needs to be pre-treated, like you are going to sterilize it and use it, because you don't want those bacteria, but you want the organic content from those bacteria that will help growth of the uncommon, unexplored bacteria, right? Now, besides the carbon source, incubation time also was very effective factor, right? What they had obtained was 110 OTUs. OTUs, operational taxonomic units. We referred to it yesterday. So a species is a very basic OTU, right? When you're talking about classification of bacteria. So they could find 110 different types, and they found out of those 110, 25 were slow growers and that exhibit significant enrichment at late incubation stage. So after 30 days, they had only about 75 to 85 of those, but remaining 25 they appeared as incubation time was prolonged. It passed about 30 days, and you could find those additional 25 new isolates which were not at all seen in first 29 days of incubation. So, nature of the complex carbon that you are choosing and the length of incubation for the enrichment media are the known things which have shown a good outcome in terms of diversity that is explored. All right. Now, this is another technique which is used, which is referred to as diffusion chamber. Now, when you talk about tapping the untapped diversity from various natural ecosystems, people have tried to simulate the natural environment. So it is another way to mimic the natural environment. So as you are seeing in the diagram, there is a O-shaped metal ring. At the base of it, there is a paraffin paper. Of course, it is sterilizable, autoclavable, that is fixed. And in the hollow part of the disc, they have poured the media, and this another plastic sheet will cover it and close it, right? That is how the disc is getting ready. Now what is put into that media? So in that media what they had done is they had added the sample may be directly from the source, or may it be the enriched medium, where the possibility of having um, more number of these desired bacteria be present. So either in the sample or directly from the source, that sample is put into the agar, and that agar is poured into this disc, and the disc is closed, and it is floated onto the chamber. Now, because something has to diffuse, from where will it diffuse? So when I'm talking about this, what you can guess is you are going to have a mini um, ecosystem generated. For example, you are tapping the marine ecosystem, and from the source you collect few liters of water as much as possible from its across the depth, right? You should have a balanced composition. So you are going to take that, bring it to your laboratory, then put it into a what we say like the aquarium, right? Aerate it, and add some nutrients to that, like glucose, etc., etc., and let the bacteria flourish. So as it goes, uh, over the period of time, you will see a scum gets formed, and slowly, because it is aerated, the scum starts getting settled onto the edges of it. Right now, you are going to float this disc onto that aquarium on the top of that water. Maybe you take it separate in a separate container, or maybe you are putting it there on the top of that aquarium itself. What is going to happen is the bacteria which are growing in that aquarium, they are producing n number of different metabolites which are not available to the bacteria if it is grown on a laboratory prepared medium. But whatever is happening under natural conditions, similar environment you have tried to mimic, a small glass chamber which is going to mimic pH, temperature, light conditions, and the water sample from the site itself, or the soil sample from the site itself you have put into it. So it is like a mini section of the ecosystem that you are going to tap, right? It is, you have created that in your laboratory. So whatever would happen in the open sea at that particular site, or the soil at that particular site, or river or well, whatever ecosystem you have chosen, to a smaller scale though, it will happen in this aquarium also. So it is like you have tried to mimic the environment in the laboratory, and you are trying to provide all those metabolites which are getting generated due to all unknown unexplored microbial activities and their interactions are available to the bacteria that would be possibly growing within this agar which is trapped in the disc. All right. So this is like this disc is going to like allow the passage of growth stimulatory chemical factors from the external environment. And because the membrane below is semi permeable membrane, so passage of nutrients is allowed, but cells are not allowed to move. So diffusion of nutrients chemicals is taking place freely across through and through, but cells are totally separated from each other. So there is no possibility of any fast growing R strategies appearing onto the disk, the gel in the disk, right? So here, of course, it is not going to be a single step process. It is to be discussed in the next lecture. I'll continue, which probably if will take place tomorrow, right? So with this introduction about diffusion chamber, we will conclude today's discussion.